The birth of single-cell Hi-C allows for studying chromatin interaction at an unprecedented resolution. Unlike bulk Hi-C data, which is measuring the average interaction frequencies of a lot of cells, it can detect variations in a single cell. Because of this variation, and because of the more complex structure of this data, currently, statistical methods for detecting biological findings of single-cell Hi-C data are scarce. No method, as we know of, tries to do integration between single-cell Hi-C and other assays. Here, we propose scGAD, which is used to better understand the connection among all these biological features. The first step is to eliminate the sequencing depth effect. After this, for each gene, we subtract the mean and divide by its standard deviation. This will allow us to remove all the biases, including GC content, mapability, and also other unknown effects. With the method in hand, we can start our data analysis. First, we obtain cell type specific marker genes defined by single-cell RNA-seq, take the average of these genes with scGAD values for each cell type, and directly visualize them using a heat map. Clearly, those cell type specific marker genes are still specific in scGAD, and this further proves the relationship between single-cell RNA-seq and single-cell Hi-C. Next, we can get to the part of projection. For the first data, 10 et al. 2021, the problem is that there are two vaguely defined cell types. After projecting it into the single-cell RNA-seq data from the same paper, we can see that most cell types are overlapping well between the two assays. What's more, if we focus on the dashed square where neonatal neuron 1 and 2 appears, there are also other various cell types from single-cell RNA-seq. If we make use of these cell types, we can successfully annotate the two cell types rather than feeling confused of what they really are. Then, still for the same 10 et al. 2021 data, we want to project them onto other assays rather than just single-cell RNA-seq. In PairTech, they measure various chromatin states simultaneously with gene expression, and we can utilize it by testing our method, to test our method on the two marks in the red box, where they have a higher sequencing depth among all the chromatin states. And it is shown here that both of them shows a successful projection. Then, for the second single-cell Hi-C data, Li et al. 2019, the problem is that subcell types are hard to separate only based on single-cell Hi-C data. We projected onto single-cell RNA-seq data from human motor cortex, and the result is shown on the right. These in the red boxes are those neuronal subcell types, and clearly they're perfectly separated comparing to the result when they're only using single-cell Hi-C. This shows that as long as there are suitable single-cell RNA-seq data or other assays, we can achieve a better cell type separation. Finally, as we all know, sequencing depth has an important role in many downstream analysis in multi-omic studies. Also, in the process of gene expression data, where what researchers usually do is to keep only top variable genes, and we're curious if this has an effect on the projection. We choose three major cell types from Li et al. 2019 randomly as subsample each cell. If we look at the heat map in the middle, in the bottom, it contains all the reads, but at the top, for each cell, it only contains 1/30 reads. From left to right, we change from keeping all the genes to keeping only 25% based on the mean value of the gene. Each entry in the heat map calculates the label transfer accuracy for each condition. And this is calculated by first building a supervised model from gene expression data, and then compare the predicted scGAD label and true label. Clearly, from bottom to top and from right to left, the accuracy decreases. This means less reads will be harmful to projection, and keeping only top variable genes are helpful for the analysis. However, another observation is that even in the worst case, the accuracy is still 0.68. And this represents the stability of projection with the change of the number of reads and genes. And that is all for the poster. Thank you for listening.